My talk is about investigating the effect of gene-country interactions on health and anthropometric traits in South Asian populations. This presentation will follow the standard format of giving you an introduction, a data and methods section, and then move on to showing my results, and then finally, a discussion. I am going to motivate my talk by first giving you some reasons why my work is important. What are gene-environment interactions, and why should we care about them? Gene-environment interactions could be defined as when the genetic component of a risk of a disease depends on a specific environment. A well-known example of this is the interaction between the gene NAT2 and smoking for bladder cancer. Why is this important? Well, one of the most important topics today are the apparent health disparities that exist within and between countries. For example, the performance of polygenic scores degrade when applied to ancestries other than the discovery populations that they were originally trained on. This is a major obstacle for population-level precision medicine initiatives, such as the Genome UK or the All of Us, as these initiatives promise equitable genomic healthcare for all. Much attention has been given to factors like the environment, linkage disequilibrium, or allele frequency differences, but another potential source of variation is gene-environment interactions on a global scale, which has been rarely considered so far. So the basic research question I'm trying to address here is how does interaction with a macro-level environment, like a country, explain health-related outcomes? The outline for this project is the following. First, identify individuals in the UK genetically matched as close as possible to those in Bangladesh. Then, find some interesting phenotypes that are common between the datasets. And then finally, fit some models like variance component analysis and GWAS to investigate G/E effects. Okay, let's move on to having a more in-depth look at the datasets and the methods I've used. Here's a slide to give you an overview for the models and the concepts that I will be using later. I assume that the phenotype is a linear combination of environment, genes, and their interaction plus some noise. This is quite close to the standard genome-wide association model, which aims to estimate the per-SNP effects. However, in situations of low power, instead of trying to estimate the individual SNP effects, either as additive or as interacting with the environment, you instead estimate their aggregate effects via so-called variance components. Variance component models aim to decompose the phenotypic variance into additive genetic, gene-environment interaction, and noise components. A core concept is heritability, which is the ratio of the genetic and phenotypic variances. Also, I have to note here that comparing or estimating heritabilities or other variance components in genetically diverse populations is problematic. So for this reason, I had to take special care to find genetically as closely matching individuals in my UK cohort as those in Bangladesh. Speaking of which, we have two metrics of genetic similarity and differentiation. These two very closely related quantities are FST and kinship. These have many definitions, but the one I prefer is to think of them as probabilities of allele sharing. Kinship is then allele sharing between any two individuals, whereas FST, or fixation index, is the same for special scenario when comparing individuals in subpopulations. This makes kinship a metric of similarity and FST a metric of difference. The lower the FST is across two different populations, the higher the kinship is and the more similar individuals are expected to be. And finally, I skipped over the interaction component. That is the heritability analog due to the interaction term, which is the ratio of the interaction term's variance component to the total phenotypic variation. Okay, so those are all the prerequisites. With respect to the data, I had access to two studies. The first was a well-known cohort of the UK Biobank, which comprises 500,000 individuals that gives us a cross-section of the UK's current population. There were around 7,000 individuals of South Asian ancestry in this dataset, as I will describe in more detail on the following slide. And the other dataset that I relied on was the BELIEVE study, which stands for Bangladesh Longitudinal Investigation of Emerging Vascular Events. This data comprised of around 55,000 individuals of South Asian ancestry in the native environment of Bangladesh. Both datasets came with genotype and imputed whole genome sequence data. After some SNP QC and InfoScore filtering, I ended up with around 800,000 common SNPs for the variance component analysis and around 5 million imputed SNPs for my G/E GWAS. As previously mentioned, to perform this work, I needed to identify genetically similar individuals in the UK Biobank as those in the Bangladeshi cohort. so the overwhelming majority of individuals in the UK Biobank are of European ancestry. However, due to this cohort being so large, there was still a substantial number of individuals from other ancestries. To identify individuals with South Asian ancestry in the UK Biobank, I took around 200,000 HFMap3 SNPs that were directly genotyped and were common between the two datasets, and then I paired from PCA. I then matched individuals on their top 4 principal components, using the criteria to select matches that fell inside the limits of the most extreme PCs of the Bangladeshi individuals. Using this method, I found around 7,000 matching individuals who achieved an FST with the Bangladeshi cohort of around 0.003. The two PCA plots here visualize how the two cohorts relate to each other. The plot on the left depicts the full UK Biobank with all ancestries combined with the Bangladeshi cohort, which shows that our Bangladeshi data, in black, falls near the individuals genetically similar to the reference populations from India, which was reassuring. And on the right, this is the final subset of individuals in our study, Bangladeshi in black and UK South Asians in the UK Biobank in blue. I chose 10 phenotypes based on availability and previous evidence of interaction effects. These traits were height, BMI, years of education, SBP, TBP, and 5 binary phenotypes, high cholesterol, MI, stroke, type 2 diabetes, and angina. In addition to these outcomes, I also used sex, age, A-squared as covariates in all subsequent models. To keep expectations realistic in terms of power, I'm showing you the prevalences of the binary phenotypes on the table on the right. UK SAS are the South Asians we have identified in the UK Biobank in the previous step. Believe are the native Bangladeshis, and GB are the Europeans in the UK Biobank, which I've included as a reference point. A quick glance at this confirms some well-known differences, such as the larger incidence of type 2 diabetes in South Asians relative to Europeans. So to convince myself that I could accurately recover the non-true effects of genes, environment, and interaction, I first performed a simulation study. I used the 6,000 individuals of South Asian ancestry in the UK Biobank and Believe, so 12,000 in total, and for this analysis, I used the 800,000 HFMap SNPs. I simulated phenotypes with the true values arbitrarily set 1 for the fixed effect environment and 33% for both additive genetic and interaction terms. To then recover these values, I fit the following model in GCTA, where I assumed that the phenotypes were normally distributed, with the mean coming from the fixed environmental effect and the variance from the sum of the genetic relatedness matrices and their estimated variance components. Now let's move on to my results. Okay, so here are the results of my simulation study, where the underlying truth included the gene-environment interaction term. So the first plot, we have on the x-axis the 3 models that I have evaluated. On the left, just the additive genetic variance, VG. In the middle, just VG plus environment. And on the right, the full model of VG, environment, and their interaction. The different colors indicate the different variance components estimated. Orange is the additive genetic, blue is the noise, green is the fixed effect, and red is their interaction. And the black bars are the true effects that I was trying to recover. As you can see, the reduced models that only included either just VG or just VG and the environment, they failed to recover the true values, whereas the full model that included all 3 terms recovered all 3 parameters without any substantial bias. The plot on the right illustrates what happens if you estimate heritability within countries, that is just within the UK or just within Bangladesh. In this case, the additive genetic variance component would get inflated by approximately the size of the gene-environment interaction term. So instead of the expected 33%, we see each VG around 66%, which is quite an interesting result in itself, because this reinforces the notion that within country heritable estimates obtained so far, they may be overestimated. This would happen if there was a hidden interaction effect of the kind I'm simulating here. As we will see, for some of the real data phenotypes, this is actually very similar to what we will find. I will now show you 2 tables of my results for the real data runs for all phenotypes, where I fit the same model as I did for the simulations I've shown you in the previous slide. The top table is the results where I combine the UK Biobank and Believe datasets into a single analysis. Each row is an estimate of a parameter of interest. H² is of course heritability, which is the additive genetic effect. Country is the additive effect of being in the UK. And the GXE is the gene-environment interaction term. In the bottom table, Because we analyzed each cohort separately, the only parameter of interest we could estimate was heritability itself, so the rows here are the narrow sense heritabilities within each study. So these two tables are the analogs of the two plots that I had shown you in the previous slide. So what can we summarize from this? We have small but significant effects for two cardio traits, SVP and TVP, and also for height. And we have two fairly substantial interaction effects for BMI and educational attainment. For edgy years in particular, my results are consistent with the whole within-country effect, basically masking a large gene-country interaction. This is supported by a bottom table, which shows that when analyzed separately, the heritability is between 0.2 and 0.3 for the two cohorts. But when analyzed together, that effect is almost entirely absorbed by the interaction term of around 0.25, with almost no additive effect. I also performed G-by GWAS using Regini, using around 32,000 individuals from Bangladesh and up to 7,500 South Asian ancestry individuals from the UK Biobank. Out of the 10 evaluated phenotypes, we found a single genome by significant locus in the case of type 2 diabetes. Looking at the Manhattan and locus zoom plus below, even this association does not look as convincing. Here is a more detailed table of the association statistics for the lead SNP for type 2 diabetes. This variant had no previous association to any phenotypes in the literature. And as you can see, the effect is quite frequent, and it was not significant in any of the single cohort or combined cohort additive association GWASs. Finally, some points of discussion. In terms of biology, the main finding of this study was that our results indicate a highly polygenic architecture of G/E effects. This was suggested by the finding of a relatively strong variance component effects combined with a much weaker signal on the per locus level. So this implies that the individual interactions are likely to be highly numerous, but with very small effect sizes. Taking a broader view, the relevance of my work is that if gene-by-environment interactions are common for diseases, then this could have important implications for treatments or drugs developed in high-income countries and then distributed across the globe. For instance, imagine a hypothetical scenario where you have performed a GWAS, which after some follow-ups and fine mapping and clinical studies leads to a drug that works, say it reduces the risk of stroke, for example. It seems to work in high-income countries, and could assume that it could work across all ancestry groups who live in high-income countries today. But then when you send the drugs to Bangladesh, it stops working. Why? Because this hypothetical drug could have been working by targeting a G/E effect, which only seemed additive in the UK. Because everyone in the UK is in one big macro environment, therefore all associations appear additive here. But because Bangladesh has a different macro environment, the target gene may no longer work the same way as it did back in the UK, which could render the drug ineffective. So the moral of the story is that G/E shouldn't be ignored, and if possible, when developing drugs or treatments, the genes that should be prioritized should be those that do not depend on a particular environment. Finally, here are a few important limitations. The main one is, of course, that with a single pair of studies, it is not possible to separate the confounding of the study itself. With a single pair of study, I could only perform gene-by-study interactions, rather than genuine gene-by-country interactions. The latter would be only possible if you had access to multiple studies. This is something that we are actively considering now to expand to utilize more studies. Another limitation was, of course, low power, especially for some of the binary traits. If we could find larger UK-based cohorts, then I think we may have been able to detect smaller effects for some of the disease traits. Next, we considered only South Asian ancestry individuals here, but there's of course no reason to expect that the effects will be restricted to South Asians. And lastly, we only considered common variants with minor frequencies over 1%, and the picture may be quite different at lower frequencies. And that is the end of my talk. Thank you. I hope that you found my work interesting, and if you have any questions, please reach out via email.